Okay, this is the third lecture in the series of lectures on the cell cycle. If I give you an overview of the last two lectures, in the first lecture we discussed about different phases of the cell cycle. Like we talk about the G1, the S, the G2 and M phase. I gave you an overall view. Then I told you that for the execution of different phases of the cell cycle, you need a machinery, and there are different components of the machinery. Now the first component of the machinery, which is the spindle apparatus, that includes the centrosome, and I've told you centrosome is made up of two things. You have got two centrioles, and you have got a mass of proteins that is known as the pericentriolar mass. I've told you that the PCM contain different kind of the proteins, like the gamma tubulin, the pericentrin and the nynine. They are responsible for a particular phenomena which is known as the microtubule nucleation. Then I told you about the microtubule nucleation process in detail, how that is going to happen, and then we discussed in detail about the microtubules, and I've told you they are actually polymers of the heterodimer of the alpha and beta tubulins. And you have got a plus end of the microtubule, which is responsible for the elongation and shrinkage, and then you have got a minus end, which is actually the response which do not have any role in the elongation or the shrinkage phenomena, but that is actually providing a relatively stable end for this polymeric structure. And these microtubules, they are of three types: the kinetochore one, the astral one, and the polar one. We also discussed about the kinetochore, and I've told you that is a protein structure which is present on the centromere of the sister chromatid, and that is actually responsible to pull the sister chromatids apart from each other. And this kinetochore actually have two subparts. One was the inner kinetochore, and the other one that was the outer kinetochore. And the last component of the machinery, that was the contractile ring, which actually pinches the two cells apart, utilizing the actin filament and the myosin 2. Today we are going to discuss about the control system of the cell cycle. If you are using a machinery, you should have control of that machinery as well. Like if you talk about the cell division, if you are using a machinery and you are not controlling that machinery, that can lead to an abnormal kind of the cell division. That usually happens in the cancer when your machinery works, when your machinery is not under the control of the cell cycle control system. Excessive cell division occurs, and that excessive cell division is usually a sign of the cancer. So for the machinery of the cell cycle, you have got a control system. This control system is made up of different kind of the proteins. The first protein, that set of proteins that is there in the control system of the cell cycle, that is known as the cell division kinase. That is also called as the cyclin-dependent kinase. Now these are the kinases which actually drive the cell cycle from one phase into another phase, and we will see that because in different phases these CDKs, they are different. In the G1 phase you have got a different kind of the CDK, in the S phase you have got a different kind of the CDK, or in M phase you have got a different kind of CDK. So these cyclin-dependent kinases are actually responsible for moving the cell cycle from one phase into another phase in a controlled fashion. If you look at the name of this particular protein, they are known as the cyclin-dependent kinases. That means that for the activity of these particular kinases, you need another protein which is known as the cyclin. Now the cyclins, they are another type of protein in the control system which actually work as the regulatory subunits of the CDKs. That means that if the cyclins, they are there, the cyclin-dependent kinases are going to work. If they are not available, the cyclin-dependent kinases, they are not going to work, and we will see them in detail in different phases how that is going to happen. Now our cells, or cells of all living organisms, what they usually do is when they need a protein, they synthesize them. when the function of that particular protein is over, and those cells are going to destroy those particular protein because their function is no more needed, and utilize the building blocks, that is the amino acid of that particular protein, for the synthesis of those proteins which are required at that particular time to the cells. So you have got a regulated destruction machinery. That means that this destruction machinery is highly regulated. That means if a protein that is required in the cell, this machinery is not going to destroy that particular protein. But if that protein has performed function and you do not need the function of that particular anymore, this regulated destruction machinery is going to degrade that particular protein. If you look at the component of this regulated destruction machinery, you have got ubiquitin protein over there. The name ubiquitin is actually derived from the term ubiquitous, because if you look at the quantity of this ubiquitin protein in a single cell, that is very high. Therefore, they are called as the ubiquitin protein. You have got the ubiquitin associated proteins. These ubiquitin associated proteins are actually responsible for the attachment of this ubiquitin protein to those particular proteins which need to be degraded. That means that the attachment of ubiquitin to a protein is actually a death signal for that particular protein. So the ubiquitin associated proteins, they are going to attach ubiquitin to those proteins which need to be degraded in the cell. When the ubiquitin that is attached to particular proteins and they get a death sentence, those are actually going to move towards the proteasome, which is actually a complex of the proteases. And you all know that proteases are enzymes which degrade proteins. So if you have attached ubiquitin to a protein and that protein move towards the proteasome, these proteases acts on that particular protein and chops it into amino acids or peptides. If you talk about the cell division overall, if you overall look at the cell division and cell growth is a highly regulated process. That means that the living organisms, the eukaryotes, they keep the number of cells in a very tight number. That means that if a cell needs 2 cells, they are going to make only 2 cells. If they need 10 cells, they are going to make only 10 cells. So the cell growth and division is a highly regulated process. If you talk about different types of the cells, like if you talk about the zygote cells, they divide every 8 minutes. Now the reason is that when the sperm and the egg, they combine with each other to form a zygote, you have to make a fetus, so you need a very quick kind of the cell division. Now the womb of the mother, that is a very nutritious environment. Growth factors, they are there. They are available in a very high quantity. So the zygote cells, they divide every 8 minutes to make a fetus. If you talk about the cells of the intestinal linings, now these cells, they divide once per day. One of the possible reason is that you take a lot of acidic food, you take a lot of basic food, you take a mixture of the foods. That means that these intestinal lining cells are exposed to a very harsh environment. So to function properly, they renew themselves, so they divide once per day. If you talk about the liver cells, they divide once per year. That means you have to take a lot of care of your liver, because if they are going to divide once per year and if your liver is not working properly, that may create a problem. If you talk about the neurons, what the scientists say that they divide once per century. Now the average lifespan around the globe is between 60 to 80 years. That means that your neurons, they are not going to divide in your whole life. But some of the scientists, some of the research in the current era do suggest that there are certain kind of the neurons that can divide. When you are dividing cells, you are not only dividing the nucleus of that particular cell, but you are also dividing the organelles of the cell, which we usually call as the cytokinases. So what is the strategy that the organelle follow during the cell cycle? If you talk about the abundant organelles like the ribosomes or the peroxysomes, now their strategy is to be abundant in the cell. Now that means that if you have got a cell over here and you have got a lot of ribosomes spread throughout the cell, and your cell divides, say for example over here, that means that both of the cells, they are going to get some ribosomes. Although say for example this cell got less number of ribosomes, this cell got a lot of number of the ribosomes. After the cell division, and these daughter cells are going to adjust their numbers. If you talk about the mitochondria, what they usually do is they divide by binary fission. They replicate their DNA and then divide by binary fissions to be abundant in the cell during the cell cycle, and they spread throughout the cell. In which plane the cell divides, both of the daughter cells, they are going to get some of the mitochondria. If you talk about the Golgi and the ER, the endoplasmic reticulum, these are big organelles in the cell, like 1 or 2 per cells. During the cell division, these Golgi and the ER are going to divide into zillion of small vesicles, and when they divide into zillion of small vesicles, they are going to achieve abundance like here. And when they achieve abundance, in which plane the cell divides, both of the daughter cells are going to get some of them, and these cells are then going to make the Golgi apparatus in ER functional after they divide. The most mysterious organelle, that is the centrioles. Like what the scientists say, that these centrioles, they are actually working like a template for themselves. As the DNA is working like a template for itself, just like that, the centrioles, they are working a template for themselves. But we do not really know how the actual mechanism, how these centrioles, they are going to divide during the cell cycle. But just like the DNA is working like a template for itself, the centrioles, they are working as a template for itself. That means if the centriole is there, you can divide them. If there is no centriole, you cannot make centrioles in the cell. Now these organelles, they are made throughout the cell cycle, but their segregation is restricted into the M phase. That means that you are dividing the organelle in the G1, in the S, and in the G2 phase, but their segregation is only restricted into the M phase of the cell cycle. We will continue this discussion in the next video.